Cancer is one of the most common and severe diseases seen in clinical practice. Early diagnosis and treatment are vital, and identification of people who are at increased risk of cancer is really important before its development. And this is a key objective of cancer research. When we talk about cancer, it's not a single disease, but a collective term used to describe forms of neoplasia. So it's a disease process that's characterized by uncontrolled cellular proliferation, and this leads to a mass or a tumor. So it's a disorder of cell cycle, essentially. Instead of being controlled, cell cycle manages to escape control, and as a consequence, you get uncontrolled proliferation of the cells, which starts the cancer process. In New Zealand, cancer is the second leading cause of deaths, and the main cancers in New Zealand that you will encounter are those that affect the lung, colon, breast, prostate, and also melanoma. There are considerable risk factors for cancer, lifestyle risk factors, including smoking, diet, environment, and particular occupations where exposure to carcinogens is an issue. In terms of diet, there can be elements that are reported to be cancer promoting or cancer protective. But the other major predisposing factor include genetics. So the process whereby a cell can manage to escape cell cycle is normally due to a mutation in a gene whose normal role is to control cell cycle. And these are typically oncogenes and tumor suppressor genes, or genes involved in DNA repair or apoptosis. The majority of cancer mutations are in somatic cells, so it makes sense. As you get older, you're more likely to accumulate more somatic mutations, which is why cancer is a disease of aging. About 1% of cancers are germline. This means that an individual will inherit a mutation or in a particular gene that's responsible for having a role in cell growth or cell cycling. And typically, if they acquire a secondary mutation, then this starts the cancer process. So individuals who have germline mutations would typically present with cancer at a much younger age. So essentially, cancer, as I said, is a disorder of cell proliferation and differentiation. So cancer cells are often poorly differentiated. So I kind of like to think of cancer as a cell that wants to not behave itself and a cell that wants to live forever. So a key goal of cancer, if you want to personify it, is that it wants to become immortal. And a way a cell can do this is by losing its differentiation. So when we try to identify a cancer cell, it's hard because it doesn't look like the type of cell that it should be, so it becomes less differentiated. It doesn't look like what it's meant to look like in terms of the population of cells that it belongs to. And additionally, it's also antisocial in that it doesn't want to respond to the signals around it, the cellular signals that tell it to control itself and to stay in check in terms of growth and cycling. So the whole reason that cancer comes about is due to this defective cellular proliferation and cellular differentiation. So cells are cycling out of control and they're becoming less differentiated, which means they become increasingly immortal, which means that the cancer cells can keep growing. They don't get recognized, and they become an immortal cell. They don't get programmed to apoptose or programmed to kill themselves. All cancers result from genetic changes, the somatic changes that transform normal cells to cancer cells. And so here we've got a diagram of cell cycle just to illustrate the process that cells go through. And genes are really important, the proteins produced by these genes, in controlling cell cycle. And they enable a cell to check, respond to signals, and either continue with cycling or to stop, or if there's a problem, even program apoptosis. So if you have a mutation in these genes that have a role in being a checkpoint, for example, TP53 is important in stopping the cell cycle for DNA repair or inducing apoptosis. So if you have a mutation in this gene, which is a tumor suppressor gene, then you're losing the capacity for the cell to check itself. On the converse of this, you have oncogenes, and their normal role is to promote cell cycle, because as a normal function, you want cells to cycle and reproduce. But if you get a mutation in an oncogene, then it pushes the oncogene out of control, and it's kind of like putting your foot down on an accelerator. The oncogene will encourage a cell to keep cycling at a faster rate than what it should do. Cancer isn't just the result of one genetic mutation or somatic genetic mutation. Typically, cancer is a long process. It's really hard to pinpoint when the first mutation occurred in the life cycle of cancer, and typically it can range from between 2 to 20 or more gene mutations that enable the population of cells to become increasingly less differentiated, increasingly less responsive to cellular signals, increasingly more antisocial, and enabling the cancer to become increasingly independent, to the point where a cancer and a tumor enables itself to vascularize. And so as the cancer grows and progresses, it acquires more mutations, and these enable the cancer to become more aggressive or to become increasingly harmful to the person. And this gives a simplification of the cancer process. So if you start off and you've got your one little genetically altered cell where the tumor development begins, and a mutation occurs that isn't checked, that isn't repaired, and is allowed to persist, and this will give that cell some particular advantage over the local population of cells. I mean, maybe it'll grow faster, maybe it will have some other benefit. So it might be that the other cells around it are in a rest state and it still proliferates, so it's growing at a faster rate. At this point, it's not causing any harm. If you look at diagram 2, you've got hyperplasia, so the altered cell and its descendants continue to kind of look normal, but they are definitely reproducing too much, and this is hyperplasia. And after years, one of these cells, so a diagram here, the pink one, acquires another mutation, and this further enables that cell to escape from the control of cell cycle. And then we get to dysplasia, and so this is where, in addition to proliferating excessively, so this population of cells now have managed just to ignore the signals around them and keep proliferating out of cycle compared to what they should do. But the offspring of the cell also appear to be abnormal in shape and orientation, and now this is when they start to look different. This is when the cells are dysplasic. And again, it could be another mutation that again alters its cellular behavior and enables it to behave increasingly differently to what it should do, and it will look a lot less differentiated than the original population of cells. We classify cancers in the term of staging and grading. Grading is focused on the morphology of cells taken from a biopsy, and cells are graded on the basis of differentiation and the number of mitoses. The more similar a cell appears to a normal cell, the lower the grade, and the grading goes from 1 to 4. Grade 1 cancers are well differentiated, with grade 4 being poorly differentiated with anaplasia. Other method of classification is the staging. This is described using the TNM classification system, where T relates to the tumour size, N to increasing node involvement, and M as to whether there's any metastasis. So this diagram is quite useful just to look at how complex, and to give an idea of the multiple factors that can contribute to cancer initiation and progression, and also just to understand that the timescale of cancer, you don't just wake up with cancer, it's something that you acquire over a long period of time. So once initiated, a cancer evolves by accumulating additional genetic damage through mutations or through epigenetic silencing of genes that encode cellular machinery that repair damaged DNA, or in genes that are meant to be controlling normality of cytogenetic behavior. The biology of cancer is very complex and multi-stage, and as I said, it takes a very long time to go from cancer initiation through to promotion to cancer progression and metastasis. And there are numerous carcinogens and factors that can initiate cancer through cell or DNA damage. And similarly, there will be multiple defense systems in our body that enable us to repair and protect against cancer. So cancer pathogenesis is a multi-step process, and it occurs because of the interaction of multiple risk factors or repeated exposure to one carcinogen, for example, in the case of tobacco smoke, in smoking. And the result is the transformation of normal cells to tumour cells, as I explained in the previous slide. So the initiation stage is the exposure to the carcinogen. Promotion is when you get that unregulated accelerated growth, and progression is when you have a malignant tumour. So understanding the difference between benign and malignant is important. Benign neoplasms are well differentiated tumours. They've lost the ability to control cell proliferation, but they're typically enclosed by a fibrous capsule, and they don't cause death unless their location disrupts the bodily function. So, for example, if there was a large benign tumour that was pushing on the brain or occluding part of the body. So you can look at the two diagrams here, and it helps to give an idea of the difference between benign and malignant cells. Malignant cells malignant neoplasms are less differentiated, they grow rapidly, they've lost the ability to control cell proliferation, but also differentiation. So they have disorganized growth, and they have metastasis, which means they have the ability then to invade and infiltrate. And typically there's two categories, solid tumors and the hematologic cancers. For a neoplasm to be a cancer, it also must be malignant, which means that its growth is no longer controlled. Tumours capable of invading neighbouring tissues or spreading, which are metastasising to more distant sites. Malignant neoplasms, as I said, are less well differentiated and can't control cell proliferation differentiation. At the point at which cells break loose from the tumour and then invade into other tissues, this is the point of metastasis, where they manage to perforate and get into the circulatory system or the blood or the lymph system and re-emerge at a new location. So key characters of tumour cells, differentiation, anaplasia, genetic instability, chromosome abnormality, and the growth. These are some different factors or different terms that all basically describing that increased antisocial behaviour of a malignant cell. So they grow independently of growth factors. They don't become inhibited by the number of cells around them, so they lack cell density inhibition. They are able to break off, so they have impaired cohesiveness and adhesion. They don't necessarily tied to the membrane, and they don't listen to other cells and what they're telling them, and most importantly, they become immortal cells. So as I said, cancer is essentially a genetic disease, so there are a lot of genes involved in cancer, a lot of genes, and it's important to recognize genetic factors in cancer because it can, in some respects, if it's at the germline level, we can identify individuals who are at a particular risk. For example, BRCA mutations, which are present in about a third of women under 29 with breast cancer, and only 2% of women who are over 70 with breast cancer. So again, illustrating like if you inherit a germline mutation, you're much likely to present with cancer at a younger age. But in addition to the germline and helping individuals identify if they're at risk, it's also important for a means of diagnosis, development of therapies, developing molecular fingerprints, and understanding epigenetic factors and microRNA genes that contribute to the progression of a cancer. And this is really important now, particularly when we're moving into the field of precision medicine. Cancers behave differently. If we can characterize a tumor, even at the somatic level, so not necessarily looking at germline mutations, but looking at somatic mutations, this can enable us to better determine treatments that the cancer is going to respond better to, that patient is more likely to have success with. So there are two key differences between cancer and other genetic diseases. Most cancer is caused by somatic mutations, and all other genetic diseases, things that we typically associate as genetic diseases like cystic fibrosis, are caused by germline mutations. So you don't necessarily pass on cancer through your sperm and eggs. Cancer is very much a disease that is acquired as a consequence of living and life. So it's often a disease associated with aging as well. And each cancer arises from the accumulation of several mutations. So it's that multi-hit process. Regardless of whether a cancer is sporadic in an individual or repeated in many individuals in a family, cancer essentially then is a genetic disease because genes are involved, and different kinds of genes have been implicated. And broadly speaking, there are 4 kinds of genes that are grouped as genes that control cell growth and replication. So we have growth promoting genes, so these are the proto-oncogenes that when mutated become an oncogene. We have growth inhibiting genes, these are the tumor suppressor genes, that are those checkpoints on cell cycle. We have DNA repair genes that have a crucial role in making sure mistakes are repaired. And then we have genes that control apoptosis. And this is an important role because if you don't have a functional process that gets rid of mutated cells, then those mutated cells are allowed to persist with mutations, and then they will accumulate more mutations. So all cancers arise because of damage to genes within the cancer. So as I said, there are these different types of genes: growth-promoting genes, growth-inhibiting genes, and death genes. So the oncogenes are the accelerator, the tumor suppressor genes are the brakes on cell cycle, and the death genes are the apoptotic genes that are important. And this is just to give you an illustration of a simplification of the steps of mutation, different mutations that are acquired that leading to colon cancer. So, and the stages in which the epithelium develops, or that cell population develops, like we had in that diagram, going from normal cell to hyperplasia to dysplasia. What happens in terms of colon cancer as each mutation is acquired in that population of cells in that tumor. And so in this case, we've got a mixture of DNA repair mutations, tumor suppressor genes, and oncogenes that enable this cancer to progress at different stages. So, and it makes sense that then the cancer is a disease that can occur over a long period of time, because you can acquire one mutation and then not notice. If someone just has a hyper-proliferating epithelium, it wouldn't present, and then another mutation is acquired. And again, a cancer won't be typically detected until it's at a much later stage, when it's actually causing symptoms in the patient. And so by this stage, many mutations have acquired, enabling that population of cells to escape cell cycle control. So with the blood cancer, again, you can look at it in three different ways, again, relating to the types of mutations that occur. So if you have, you can either look at a blood cancer as proliferation, so a mutation that enables a population of cells to just grow and grow and grow, so you get population growth. You can have mutations that mean that cells are unable to grow up, so they have the inability to differentiate. So it's kind of like being stuck as a teenager for the rest of your life. And so you either get baby cell overcrowding, which are the blasts, or you get teenage cell chaos, so in promyelocytes. So you've got, instead of the cells progressing through that stage of differentiation, they get stuck at a particular age. Or the other type of cancer results as the cells have an inability to die, so geriatric overcrowding, so in the case, for example, of lymphoma. There are gene mutations specifically associated with different types of blood cancer, which you've probably covered already. For example, we have the FLT3 mutation, which is present in about 30% of acute myeloid leukemia cases. This is an important cell surface receptor in developing blood cells, so normally it needs to be activated by a protein, and then the dimerization of this protein enables a cascade of signaling to occur. But if you have a mutation, then it's kind of switched on the whole time. It's a tyrosine kinase, so it regulates cell survival, proliferation, and differentiation of stem or progenitor cells. So mutations enable this to be cause it to work all the time. So it doesn't need to have a signal in order to be activated. So it switches this accelerator kind of on without needing another protein to switch it on. JAK2 is another important gene with hematological cancers. The mutated forms, if someone is JAK2 positive, then it doesn't need EPO as a signaling molecule, it's always active. So again, it's a kinase, and it's misregulated or mutated in a number of myeloproliferative diseases. The V617F is the most clinically relevant variant, so the one that you'll most likely encounter, and seen in about half of myeloproliferative disorders. If it's a mutated form of JAK2, it binds and triggers hyperactivation that promotes cell growth and production. So its normal role is to provide instructions for making a protein that promotes growth and division, and it's part of a signaling pathway. And so if you have a mutation in it, it's kind of switched on the whole time and doesn't need to receive a signal to be switched on, so it's always active, so you get constant cellular proliferation. Another example from the hematological cancers, with follicular lymphoma or non-Hodgkin's lymphoma, you get translocations that bring two genes together that shouldn't really be together, and so you get fusion genes that are hyperactive. In the case of non-Hodgkin's or follicular lymphoma, it's a characteristic 14-18 translocation, which puts the BCL2 gene on chromosome 18 next to the IGH gene on chromosome 14, and then you get overexpression of the BCL2 protein, which is its normal role is an inhibitor of apoptosis. So again, the tumor cells then become immortalized. So if you have overactive, so BCL2 blocks cell death. So if you have overactivity of BCL2, you're not going to have any apoptosis. So you get that geriatric overcrowding. It's common. It's a common cancer, slow growing, typically in adults in 60 to 65, and the cells build up in the lymph nodes. Another translocation is the Philadelphia chromosome associated with chronic myeloid leukemia. It's a 922 translocation, and it makes a novel protein. And the novel protein is a tyrosine kinase, so again, another promoter of cell cycle, another oncogene, and it's always active. So having this fusion gene means it's always active. It's present in about 95% of classic CML patients, and it results in the proliferation of granulocytes and their precursors. So on the chromosome 9, the Abel gene is a tyrosine kinase, and the BCR again is that signal regulation. So the fusion protein enables the cells to grow and divide, and blocks apoptosis of abnormal cells. So it's always switched on, so the growth is uncontrolled. And this is an example of a drug that's been designed to target specifically CML by targeting the BCR-ABL fusion gene, imatinib. It's a biological. It's one of the early smart drugs. It's a tyrosine kinase inhibitor, so it's specifically targeted at these tyrosine kinases. Here's another fusion gene. So in acute promyelocytic leukemia, you have a 1517 translocation, and this results in the proliferation of promyelocytes. So again, this is like a build-up of the teenage cells. They can't differentiate. They're not able to grow up. And it's rapidly lethal if untreated, but it has a higher cure rate if treated. The fusion gene is the primary abnormality that leads to the acute promyelocytic leukemia. It involves the Ra alpha gene, which is involved in gene transcription, and the PML gene, which I think is a tumor suppressor gene. So it produces a fusion protein that interferes with normal function, and blood cells are kind of stuck at this promyelocytic stage, and so they proliferate in an abnormal rate. In terms of future, so there will be more DNA and RNA-based diagnoses using next-gen sequencing and gene profiling. So we at the moment are using the Illumina hotspot panel, which looks at sequencing 50 tumor suppressor and oncogenes as a way of characterizing different cancers. Because if we understand different tumors or different cancers, then we can better understand how to treat that cancer and what type of treatment it will respond to, or design biologics or specific treatments that are targeted at that particular molecular pathology. So it enables us to identify further molecular abnormalities and increased diagnostic precision for more sensitive disease monitoring. So there's likely to be increased use of molecular and sequencing data in monitoring therapy and designing specific therapies. It also is likely that there will be new future treatments will be drugs that are designed specifically towards those molecular defects. So, for example, as I mentioned, the imatinib, the tyrosine kinase inhibitors designed specifically against inhibiting the defect as a way to move away from chemotherapy. And there will be new biological agents that incorporate viruses, viral vectors, monoclonal antibodies, chymogene antigen receptors as more effective targeted treatments with less side effects. There will also be experiments using, obviously, gene therapy, particularly for single gene disorders and some of the hematological tissues, and a lot of interest in genome editing using CRISPR-Cas9, which is a directed type of gene therapy. So you get a small piece of RNA with a short guide sequence, and it attaches to a specific target sequence of DNA in the genome. And the RNA also binds to the Cas9 enzyme. And as in the bacteria, the modified RNA is used to recognize the DNA sequence, and the enzyme cuts DNA at the targeted location. And when the DNA is cut, uses the cell's own DNA repair machinery to stick in or delete a piece of genetic material to make a change. by replacing an existing segment with a customized sequence as a way of correcting problems in particular cell populations. So we're there yet, but that's something potentially in the future that will be increased more in the treatment of cancer.